By understanding DNA and cell structure, scientists have developed several techniques to apply this knowledge. This video looks at three of these: genetic modification, artificial cloning, and the use of stem cells. Firstly, let's compare genetic modification and cloning. GM is where a single gene is moved between species, allowing one of them to make a new protein. Artificial cloning, on the other hand, is producing a genetic copy of an entire individual so that they share identical genomes. Because all living things use DNA in the same way and use genes to produce proteins in the same way, it's possible to take a gene from one species, insert it into another, and for it to work. This can take place between any two species. The diagram shows how the human gene for insulin can be cut out of a chromosome and added to the DNA of a bacteria. When the bacteria cell divides to reproduce, each new bacterium contains the copy of the human insulin gene. Each bacteria is then able to make or synthesize the insulin protein, and when it's extracted from the cells, it's identical to the insulin you would collect from a human. This is how the insulin needed to treat type 1 diabetes is made. This way of producing insulin is cheaper and more reliable than other methods. Using a similar technique of cutting out a target gene and inserting it into a new species, the gene for beta-carotene, which is needed to make vitamin D, has been added to rice. This is because areas of the world where rice is a staple food were noticed to be suffering from high levels of cataracts. This is an eye disease caused by too little vitamin D. The idea was to make rice produce what is needed to make vitamin D to solve this problem and improve the health of many millions of people in this area of the world. When growing any crop plant, one of the things that will limit how much you grow and therefore how much profit the farmer makes is weeds. You can spray a herbicide on the crop which will kill the weeds, but this is also likely to damage the crop as well. By inserting a gene that causes some plants to be naturally resistant to the herbicide into the crop, you can then spray the field to take out the weeds without damaging the crop. This makes it easier and cheaper to produce more food. Unfortunately, whilst all three of these uses of GM are positive, things are rarely that straightforward. There are ethical considerations in moving genes from one species to another, particularly when the genes are human. Golden rice is more expensive than normal rice, which makes it inaccessible to the poorest people who are those most in need. You also need to consume a large volume of the rice to be able to produce high enough levels of vitamin D. With the herbicide-resistant crops, there's a risk of the gene for resistance getting into wild populations and producing superweeds that cannot be treated by the herbicide. Moving on to look at artificial cloning, this is where instead of moving a single gene from one species to another, an entire copy of one individual is made. The copy is genetically identical, meaning all of its DNA is the same. This diagram shows how an animal could be cloned. The green cell is taken from the animal you wish to clone. The nucleus, which is the part of the cell containing the genes, is removed. This is called enucleation. An egg cell from an individual of the same species is collected and the nucleus removed. The chromosomes from the egg will not be needed. To get the nucleus from the cell you are copying into the empty egg cell, a small jolt of electricity is used. You now have a single cell where the DNA is identical to the one taken from the animal being cloned. No DNA remains from the animal the egg came from. Another small jolt of electricity causes this cell to start undergoing mitosis to grow, forming an embryo. The embryo is then implanted into a female, which will give birth to the clone once fully developed. This process is beneficial when you have an individual with a set of characteristics that are really useful. For example, a cow that produces a lot of milk, or a racehorse that's particularly fast. By cloning rather than breeding, you can guarantee that all the genes from the original animal will be passed on. It could also be used in the conservation of species that breed really slowly, to build up populations quickly. There are several arguments against the use of artificial cloning, however. The process has a low success rate, and results in some embryos being destroyed before they're ready to be implanted. There's a risk in producing a population with very little genetic variation. For example, if one individual is able to contract a disease, then they all will. Before being born, artificial clones sometimes grow too large, which puts them and their mothers at risk. Sometimes the internal organs are too large as well, which affects health. Artificial clones can also age quicker than normal animals, reducing the lifespan of these animals. One potential use of cloning is producing organs to use in transplants. A better way of doing this is developing through the use of stem cells. Stem cells are cells that are undifferentiated, which means they have not specialized to become a particular type of cell. Adult humans do not have full stem cells, but other animals that are able to regenerate body parts, such as lizards and spiders, do. Humans do have stem cells as embryos, and there are some present in the umbilical cord that could be collected at birth. Using these stem cells, doctors could potentially use them to grow specific groups of cells that could help treat diseases such as Parkinson's. They could rebuild bones and cartilage in people with degenerative disease or who have suffered severe injury. They have also been used to grow replacement valves to go inside hearts when these have failed or become damaged. In summary then, genetic modification is the process of taking a gene from one species and inserting it into the genome of another. Because every living thing uses DNA to make proteins in the same way, this gene will work. Genetically identical individuals can be produced through artificial cloning, although the health and ethical risks of this are significant. Stem cells, which are undifferentiated cells that can be manipulated to become any type of cell, could be used to grow replacement body parts for people suffering injury or disease.